Hello friends, myself Dr. Aashish Sharma and I welcome you all in my YouTube channel Concepts Connect. Before I move further, I would like to let you know that this video is the learning outcomes of my last 12 years experience in the field of biomaterials formulation development. During these 12 years, I have been associated with biomaterials field as a PhD scholar, postdoctoral researcher, and now I am working as an associate professor guiding masters and PhD students who are working with biomaterials related research projects. Being a PG and PhD supervisor and a postgraduate research coordinator in the School of Medical and Allied Sciences, GD Goenka University. I felt that most of the research scholars are struggling badly with designing and execution of their research projects, and most of their research ends with a substandard publication only. To help them out, I prepared this video, Meticulous and Ingenious Approach for Biomaterials Research Go Beyond Publications. I can make this title easier for you as Approach for Developing Biomaterials Formulations. So I request to all the young researchers, please watch this video till the end and multiple times for better understanding about biomaterials research projects. I believe you all would surely be benefited. If you feel it is worth watching this video, please like this video, share among your friends, and more importantly, subscribe this channel. Let's talk about research dreams or research goals of fresh masters and doctoral students. Most of the young researchers dream for a patent or developing a new product, but while execution in the middle stage of research, they struggle with multiple problems such as unable to identify suitable research problem, superficial literature search, improper research planning as per timelines, cost assessment, no idea about design specifications, scalability of research at industrial scale, lack of knowledge about market competitors. I believe most of the master's students and early PhD students stuck with these problems, and at the end they have to be satisfied with a research publication only, sometimes with substandard publication. Don't worry, here I am going to discuss solutions of all these problems. So keep watching this video till the end. I strongly believe that after watching this video, you can handle the above mentioned problems effectively and will surely have the wonderful outcomes of your research more than publications. As we all know that majority of the research project aim to IP generation, licensing, funding for startups, application for US FDA and European Union certification or similar things. Every researcher must aim for these goals, but achieving these goals can be doubtful or sometimes not possible if you are not working on right research problem. So your research problem is the foundation and fuel as well, which support and drive your research journey and helps to achieve the desired research outcomes and your research aims. Once you finalize the right research problem, you can go ahead with your literature search. Although your literature search must have started quite earlier to find out suitable research problem and should be continued during your entire research project. Alright, after finalizing your research problem, the next step is to form a perfect team for efficient execution of your research project. Here, you should notice that finding a research problem must be a first step because research problem only will give the direction of your literature search as well as will let you know about what type of skill set or team members you need to complete the research project. I think you are understanding me. Alright, let's move ahead. Next step is to develop design specifications of your product, which will help you to decide what type of biomaterial formulation, for example, hydrogel, nanoparticle, etc., you have to develop to solve the research problem. Next step would be selection of base polymers and cross-linkers. Then you can go ahead to develop your biomaterial formulation which can satisfy your design specifications. Whether your formulation meets the design specifications of the product or not, this you will know by this suitable characterizations. Development and characterization of formulations. These steps may have to be performed multiple times to get best formulation which can satisfy your design specifications. If all these steps are performed carefully, critically and by heart, believe me, you will surely have better chance to generate an IP, license, funds for startups, etc. In the remaining part of this video, I will explain each step in details. We have talked that successful, tangible and monetary outcomes of your research largely depends upon research problem. During interaction with postgraduates, students, including PhD scholars, I found that they deliberately target some publications only as their research outcomes. It is not the case that they don't want to have patents or commercialize their products. Yes, they want to see their products in market, but due to unnecessary rabbit race of getting publications and poor strategies of research, these early career researchers are bound to live with publications only. There is no harm in getting good publications. Every researcher must get good publications, but we should always think some better outcomes along with publications in terms of commercialized products or startups. Let's talk about the current trend in research students to find out research problem. Or I must say, they are finding the way by which they can publish a research paper only. So most of the early researchers start finding research problems by reading research articles only. While doing so, they get involved inadvertently or unknowingly to find out what others did not get published so that they can perform or develop the same to get a publication, and during this exercise they forget the true research problem. In nutshell, they read research articles to find out what others did not get published to publish another article. In my view, this is a wrong practice of finding a research problem. Here I am giving two examples of finding out research problems in biomedical field. To find out a suitable research problem, you may focus on such problems which affect a large population of the world. It may be a healthcare problem badly affecting a huge population. I am taking an example of cancer badly affecting the entire world. In this case, you must check the number of cancer cases expected to grow in the near future in 10 to 15 years. Now, you yourself can think whether the treatment of cancer is more important or just a publication. Yes, of course, treatment of cancer is more important than thousands of publications. You will anyways have good publications after finding treatment of cancer, but the treatment of cancer you develop will always have enormous life-saving and monetary potential. In this case, your research problem may be to find out cause of cancer, find out ways to target cancer cell imaging, and so on. Another example of research problem can be adhering and sealing pulsatile and wet human tissues, as adhering living tissues in wet environment is one of the biggest challenges. The solution of this problem may come out with development of surgical adhesives or sealants which have huge healthcare and monetary potential and benefits. You always must check the market growth in the field which is associated with the concerned research problem. You can see in the bottom right of the slide, I mentioned 10 years market forecast or market growth in the field of surgical sealants and adhesives. Now you know the research problem, adhering wet tissues and its future demand and market forecast. It clearly indicates that more investment will come into this field and more chances of funding will be there. More healthcare industries will be willing to start research and development and subsequently production of surgical adhesive and sealants. Now I think you can realize the scenario clearly. If you choose the right research problem, you will automatically improve your skill set to handle the research problem. And because the research outcomes or product are more demanding, your research can get the attention of scientific community easily, and you will have more chances of getting startup funding and job opportunities. So now you know how to select the right research problem and what are the benefits of choosing the right research problem. Having chosen the right research problem, now the next very crucial step is to constitute a perfect team to have the desired skill set to execute the research. Building a right team is always necessary because solving out a research problem or developing a product for solving a research problem, you may need different expertise and skill sets. Here, I am taking examples of two research problems to explain team building. First example is development of surgical adhesive for femoral artery closure and its healing. Let's see what sort of skill set we require for developing a surgical adhesive for femoral artery closure and healing. The first person would be, of course, biomaterial scientist who will deal the polymers to develop adhesives or sealants and characterize the same. As you are developing adhesive or sealants for femoral artery closure, the person who is going to use these adhesive in real time on patients would be a vascular surgeon only. Moreover, vascular surgeons know femoral artery in and out and are needed to assess the clinical applicability of adhesives. Another team member may be a device engineer. A device engineer can contribute to develop a device to deliver the adhesive based on the inputs from vascular surgeon and a biomaterial scientist. You will need a regulatory or an IP expert also for regulatory or IP processing. You must also need a biologist to perform biological studies. If the entire team will work in harmony, there would be more chances to commercialize your biomaterial formulations. I am taking another example of team building for development of multimodal nanoparticles for cancer treatment and imaging. First team member would be a biomaterial or polymeric chemist to develop and characterize nanoparticles. Cancer biologist may be another team member you require to take care of biological experiments. Oncologist will contribute to clinical aspects of cancer therapy and management and give feedback to biomaterial scientists and cancer biologists to improve the formulation. Finally, you will need a regulatory or IP expert for regulatory or IP processing. Okay, now we know that for successful execution of research project, we need to have right team. Another very important step of developing a biomaterial formulation is framing design specifications. Firstly, let me tell you what are the design specifications. Design specification is a written document that explains your product or biomaterial formulations such as hydrogel, nanoparticle, bioadhesives, and maybe a tissue scaffold. Design specifications specify what you want your biomaterial formulation to perform. For example, you are developing a bioadhesive for sealing the femoral artery or developing nanoparticles for targeted cancer therapy and imaging. Design specification also tell us that how the user of your biomaterial formulation should interact with it. Citing the above mentioned examples, the biomaterial formulation, for example, it may be a bioadhesive or nanoparticles, must show patient acceptability, easy to be used by a doctor and easy to formulate by a scientist as well. Note down the design specifications is the most crucial step must be done in the early stages of product design and development. The design spec or design specifications direct the whole scope of your product development. How design specs are decided, I will explain in the coming part of the video. Here, I am going to tell you what points must be considered for developing design specifications. I am taking an example of developing a surgical adhesive or sealant for femoral artery closure and its healing. We start with expected functions of biomaterial formulation which is supposed to perform. In the above mentioned example, the proposed adhesive must seal and heal the artery. For performing the desired task, the adhesive must have the following properties such as fast curing, resist high blood pressure, can work in presence of blood, helps in artery regeneration and must be biodegradable and biocompatible. Now we move to target site's properties, which is femoral artery in this case. Whatever product we are developing, it must be compatible with the tissue we are applying the formulation upon. Here, adhesive must be compatible with femoral artery properties such as physiology, composition, elasticity, pulsatile frequency, and regeneration time of femoral artery. Knowing the properties of femoral artery or target site will help us to tune the essential properties of the adhesive according to the target site so that the adhesive can perform the desired function. Now we can think what type of biomaterial formulation you have to prepare so that cuts in femoral arteries can be sealed immediately and effectively. And your formulation must support tissue regeneration also. This means you may select a suitable biomaterial formulation such as hydrogel, film, nanoparticle, etc. By this time, you might be knowing expected functions of your biomaterial formulation, properties of site of application, and type of formulation. Having known these things, one should move to select different constructions material for developing a biomaterial formulation so that you can develop a biomaterial formulation with desired design specifications. Literature search must be done accordingly to select suitable biomaterial formulation, polymers, crosslinkers to achieve the design specifications. Experiments must be designed accordingly, laboratory animals should be selected accordingly, and market competitors must also be evaluated accordingly. In general, there are four aspects which must be considered before developing a biomaterial formulation. First one, where we want to use the biomaterial formulation and what purpose is it going to be used for. For example, we must think whether we are going to use the desired biomaterial formulation for lungs, heart, blood vessels, or gastrointestinal tract. Then only we can think about the type of biomaterials formulation. For example, for drug delivery to lungs, nanoparticles may be the most suitable biomaterial formulation. Adhesive patch looks suitable for sealing cardiac defects. In situ hydrogel seems okay in case of arterial anastomosis. Tablet or capsules or nanoparticle solutions look fine for gastrointestinal delivery of medicaments. The second important aspect is scalability of the proposed biomaterial formulation at commercial scale at affordable cost. Formulation of biomaterials should not need any special reactor or device or production line. This may be one of the ways to make the biomaterial formulation affordable. The biomaterial formulation must be designed ergonomically so that it can be used easily by all its users. For example, a surgical adhesive or a medical device will be used by a patient, doctor, device engineer, and a scientist. We should always think about better post-application experience of biomaterial formulation by different users, mainly patient, for example using a surgical sealant. An example of biomaterial formulation may reduce patient's stay time in hospital after a surgery. In this video so far, I have talked about approaches for identification of the research problem, team building, and developing design specifications. Now I will let you know how you can select a biomaterial formulation and its polymeric components to match the design specifications. I am taking an example of development of surgical sealant for closing and healing femoral artery. As we have already talked that we must keep the properties of application site or properties of femoral arteries in this example into our mind, such as elasticity, pulsatile movement, modulus, regeneration time, resorption, etc. If we think about all these properties, we will realize that we need a biomaterial formulation which has spread ability on artery surface and can make bond with outer surface of the artery. This proposed formulation must have comparable elasticity with the elasticity of femoral artery. This formulation must be flexible and act as a porous scaffold after curing. Although we know lots of biomaterial formulations such as nanoparticles, films, scaffolds, layer by layer assemblies, biodegradable sutures, but most suitable biomaterial formulation which can satisfy all the design specifications may be a hydrogel. Yes, a well-designed hydrogel may possess suitable spreadability over the artery surface, tunable elasticity and flexibility, and may have porous nature even after curing. Chemical functionalities of polymers of hydrogels can also make bonds with outer surface of artery. None of the biomaterial formulation, including nanoparticles, films, scaffolds, layer by layer assemblies, biodegradable sutures, can have all these properties. Hence, we can choose hydrogel as a suitable biomaterial formulation over other formulations. Now we have to think critically about the polymeric components of hydrogels, because physicochemical, biological, mechanical properties and cohesive and adhesive strengths of biomaterial formulation, in this case hydrogel, largely depends on its polymeric components. If we are proposing an adhesive hydrogel for closing and healing femoral artery, the polymeric component must support this hydrogel to attain the desired features like elasticity, fast curing, spread ability, bonding with artery surface, and flexibility. Along with it must be biocompatible and biodegradable. So, the base polymer must have chemical functionalities for easy chemical derivatization or cross-linking, and RGD sequence for promoting tissue regeneration, because we want to regenerate the artery as well. This polymer must be elastic, biocompatible, biodegradable. In this polymer, we always want to see some properties like it must be already appeared in FDA or it has the FDA history as implant. It would always be better in this case if it has the FDA history as implant, because if you want to commercialize your product, it would be beneficial. So, as a holistic approach, you must review the properties of different polymers and then you can choose the suitable polymer for formulating adhesive hydrogel. Among plethora of polymers, we can choose gelatin, which have all the above mentioned properties to formulate an adhesive hydrogel to seal and heal artery. Gelatin and hydrogel were just examples for explaining how we can select a polymer and a biomaterial formulation for particular application. Another important thing you must notice that selection of polymers and biomaterial formulation depends upon site of application and the purpose. By this approach, you can select a polymer to develop the desired biomaterial formulation as per the application and design specifications. You people can try this approach and can contact back to me if you have any problem. I hope many of you understand how we can develop design specifications. If not, no problem. I'm explaining approach of developing design specification again by taking another example of development of multimodal nanoparticles for targeted cancer therapy and imaging. As we did earlier, we start thinking from application part or functioning. In this case, main application of biomaterial formulation is targeted cancer therapy and imaging. So, cancer imaging agent must be long-lasting, proper visualization must be from deeper tissues, on-demand removal of imaging agent from human body, imaging must be selective to cancerous cells only and non-toxic to healthy body cells. Formulation must target cancerous cells only. Multimodal nanoparticles must be biodegradable and biocompatible. Keeping all these specifications into our mind, we will develop multimodal nanoparticles decorated with imaging agents, ligands for cancer cell targeting, and these nanoparticles would be able to deliver the desired amount of anti-cancer agent into the targeted cells. In nutshell, multimodal nanoparticles must be developed in such a way that it must fulfill the desired specifications. Once we note down the design specification, we can select polymers, cross-linkers, and imaging component accordingly. Experiments must be designed accordingly. Experimental animals should be selected accordingly. Literature search must be done accordingly, and market competitors must also be evaluated accordingly. So, I believe you people can understand the approach to develop the design specification. If not, kindly watch this video twice or thrice for better understanding. Having finalized design specifications of a biomaterial formulation, it would be very easy to select suitable polymeric component and biomaterial formulation. This might be the right time when you can do prior art search for any published or granted intellectual property or patent. You can use United States Patent and Trademark Office and Google Patent web portals for this purpose. Simultaneously, you must look into the characterization part of your biomaterial formulation. Polymer and formulation characterization in vivo and in vitro studies must be performed as per standard guidelines such as ISO, American Society for Testing and Materials or ASTM, and FDA, etc. Next important parameter is the financial planning. Costing of chemicals, instruments, characterization, animals, approvals, and IP processing must also be calculated precisely. Having done all these exercises, your biomaterial project is ready to execute now, which starts from first step, which is literature search and preparation of design specifications. Second, polymer synthesis and characterization. Then third step is biomaterial formulation and development. Fourth step must be prototype development, testing and iterations. If you planned well your project and work hard for its execution, then there is a good chance to generate IP, licensing, spin-outs and startups. Here I use a term prototype. Let me tell you what does it mean. It means the first model or design of something from which other forms will be developed. In our case, the prototype would be first biomaterial formulation for testing its function. That's all in this video. These things sometimes take time to be understood. So I suggest you all to watch this video twice or thrice if needed, so that you can understand the process of developing the biomaterial formulation in a better way. If you like this video, please share among your peers and research scholars and students. Please subscribe this channel for getting these type of educational videos. Millions of thanks and regards. See you soon with my next video. Have a nice time.